Hello everyone, hope you all are doing well. So today we are going to discuss the next question of our agriculture optional answer writing practice series, and the question for today is briefly discuss the biotechnological approaches in improving yield and quality of wild. So we will see how we can answer this question. So it is generally about biotechnological approach. It doesn't matter either they give here oil or grain or anything, cereals, pulses. You can all you can use the similar approaches in all the answers, but you can provide a specificity to your answers by providing examples. Suppose here the question is on improving yield and quality of oil. So in examples you will provide examples related to while when they ask here, suppose for example about pulses, so in example you will add pulses. If they ask about something any other crop, so you will use the examples of that crop in the generic statements or generic points, so enhance specificity in your answers because the approaches are going to be same only. So let's see how we can approach the answer. So we all know what biotechnology is, that is involving of biology plus technology. You have all heard about various crops, for example, BT cotton. So it is a kind of example of biotechnology only. Without technology, BT cotton would not be possible today. And using this biotechnological approach, you can enhance quality, you can enhance stability, you can enhance quantity, you can enhance yield, that is quantity, you can enhance quality, stability, everything in the crops. Okay, and this is how you can introduce. You can write something about biotechnology, and you can give examples of some wild seed crops such as soybean. Then we have rapeseed. Then we have groundnut. Then we have sunflower. All these examples can be given. Okay, then we will move to approaches. So what are those biotechnological approaches which can help in improving yield and quality of wild seeds or wilds? First one will be the genetic engineering. Genetic genetic engineering. So in genetic engineering, there I will divide into two three type parts. First one could be the overexpression of key enzymes. Through genetic engineering, we can overexpress the key enzymes. Overexpression of key enzymes. You can give examples of some enzymes such as which can enhance oil quality. For example, acetyl CoA carboxylase. Acetyl CoA carboxylase. You can give examples of fatty acid synthesis, fatty acid synthase. So these are the enzymes which can enhance the kind of wild content uh, in the wild crops, and so this process would be the over expression of these enzymes which can enhance the yield. Secondly, we can introduce genes from other species. Introduction of genes from other species. So for example, you can mention here about we can insert lauric acid producing genes from lauric acid producing genes, okay, from California bay into canola. From California bay to canola. And what happens, this due to this lauric acid uh, from California bay, the lauric acid content of canola can increase, enhancing its quality. You can also mention here about RNA interference. RNA that is RNA interference. So it can deregulate some undesirable fatty acids also. For example, uric acid in rapeseed. So you all know this uric acid. Uric acid is not that desirable and generally in low, it should be in low content. So in rapeseed through RNA interference, it can be reduced or down regulated. So these are the things which we can do through genetic engineering. So we can overexpress certain enzymes which can enhance the yield. We have given examples. We can introduce genes from other species. For example, we can introduce lauric acid from California bay to canola to increase the lauric acid content of canola and overall increasing the quality of the oil or quality of canola. Okay, then we can do RNA interference to deregulate or down-regulate some undesirable fatty acids. For example, uric acid in rapeseed. So these things can be mentioned. Secondly, we can use molecular assisted selection, MAS, that is molecular assisted selection. So anyway, we can use molecular markers which are linked to high yield and favorable fatty acid. For example, we can use molecular markers for high oleic acid or reducing or lowering linoleic acid, linoleic acid, linolenic acid. So these things can be done, and this can speed up the traditional breeding program. So there are various traditional breeding programs. The MAS, that is marker assisted selection, will speed up those traditional breeding programs also. We can use tissue culture and soma colonal variations. Tissue culture and soma clonal variations. So here we can use, or you can select the soma clones with improved traits. We can micropropagate of phyllite genotypes with superior oil content. So obviously we can do tissue culture so that we get superior wild types. We can use CRISPR Cas9 technology, that is used in genome editing. Genome editing. So here we can do precise editing of genes involved in fatty acid metabolism to enhance viral quantity and tailor composition. We can enhance the composition. For example, we can knock out or remove FAD, FAD2 genes to increase oleic acid content. And what it will do, it will increase oleic acid. We have mentioned here oleic acid content. So this can be done. Oleic acid content. We can also do metabolic engineering. Metabolic engineering. So here we can manipulate entire biosynthetic pathways. For example, we can increase triacylglycerol synthesis. It is also known as TAG, triacylglycerol synthesis. We can improve wild accumulation in non-traditional tissues. Okay, for example, we can improve wild accumulation in leaves. We can improve wild accumulation in stems, which can be helpful. Okay, next we can use transgenic approaches. Transgenic approaches, and these approaches will enhance stability. For example, a plant which is resistant to kind of wild density, which can improve its resistance, increase its shelf life, increase its usability, and is resistant to rancidity. So all these things will what do? It will enhance the quality of oil only. Okay, we can do bioprospecting and gene mining. Bioprospecting and gene mining. So we can through gene mining and bioprospecting, we can go deep into the wild varieties. We can go deep into wild relatives. We can go deep into microbial sources to get some novel genes for wild traits which can be introduced into commercial crops. Okay, so here we can use wild relatives, we can use microorganisms. So anywhere from anywhere, if we are getting some good genes to enhance the wild quality and content of wild crops, we can use it. So these are the 6-7 methods of technology which can be used to enhance quality and yield of wild. So I hope you understand it. In conclusion, you can mention biotechnological tools, especially genetic engineering and genome editing, offer precise and efficient methods to improve wild seed crops for higher yield, better quality, industrial stability, and contributing to food security and health. So here we can do all those things with higher precision, which can be mentioned in your conclusion. So I think if you write all these things, these would be enough for your answer. We have discussed, I think, 7 approaches which can be mentioned in your answer. So that is all for today. If you like it, please press the like button. Please do share and please do subscribe. It takes a lot of hard work to make such videos for you on regular basis. And if you like, if you share, if you subscribe, it motivates us to make more such videos for you regularly. So that is all for today. Have a nice day. Thank you.